In this protocol, we use 3 different DNA samples: a control non-TGO salvia, a TGO positive control, and curcuma DNA extracted from last practical. Keep always all the tubes on ice to prevent enzymatic activity. First, take a sample from the salvia DNA tube. Then, place 5 microliters of the salvia sample on 2 plastic tubes of 2 milliliters each.
In plastic tubes of 2 milliliters, place 5 microliters of curcuma sample. In the other plastic tubes of 2 milliliters, place 5 microliters of transgenic organism control sample. Add 5 microliters of the plant master mix, the green one, on the corresponding tubes. Add 5 microliters of the GMO master mix, the red one, in the corresponding tubes. As you can see, we have 2 tubes per sample. On the left one, we added the plant master mix, and on the right side, we added the GMO master mix.
For this experiment, you will set up 2 PCR reactions for each DNA sample, which makes 6 PCR reactions in total. The plant master mix was used to determine if the DNA extractions of the curcuma was successful by the identification of a DNA sequence that is common to every plant with green-colored primers. This amplifies the photosystem 2 used in the light reaction. Your DNA sample contains GM DNA sequences. With specific red-colored primers in the GMO master mix, common DNA sequences of GM plants are identified. A non-GMO control is needed to know if the reactions have been contaminated by GMO-positive DNA.
Once in the PCR, the DNA is denaturated at 94 degrees Celsius for 2 minutes for 1 cycle. Then it is amplified, beginning with a denaturation at 94 degrees for 1 minute, repeating 40 times, followed by annealing at 59 degrees for 1 minute, extension at 72 degrees for 2 minutes, and a final extension at 72 degrees for 10 minutes, 1 time, followed by 4 degrees, an indefinite amount of time in order to preserve the DNA. Does that make